Thank you, and good morning. The first point I would like to make is that I'm definitely no specialist. The whole area of GMO is so broad from the scientific point of view into social aspects of the GMO use and into juridical specificities, and so no one can really be a specialist in GMOs. What I can say is that I am a biologist by training, and I worked 7 years in a green organization working with rainforest and campaigns of all kinds of stuff. And the last 18 years I've been running my own consultancy, where I help mostly organic companies and organic organizations here in Denmark in developing their business. So it's business plans and it's project proposals and stuff like that I'm doing for a living. And then I also do some you could call them technology assessments. So if a new packaging material appears, a company will ask me, well, is this good or is it bad? Should we use it or shouldn't we use it? And if we use it, what should we consider? And so I do those kinds of assessments for companies. And 4 years ago, the Danish Minister of Agriculture came out with a white paper on GMOs. And what it said was that there may be small risks, however, we handle them, all of them. So really, it was a payment for going forward with GMOs now in Denmark. And the minister herself had been visiting Monsanto, and it was a really good company. And I would say when I read this report, and the more I read, the more I think this is shit. And so I started digging down to it. And one thing that I found was that even though there were a lot of resentment towards GMOs, the amount of documentation that showed that GMOs is really not good, it was very scarce. There are very few scientific investigations that show that there really is a problem. And it was only later I learned that the companies, they can themselves decide which scientists are allowed to do the research. So the ownership of the seed makes it impossible for interested scientists to make research. You know, when we in another area we have a lot of patents is within the area of medicine. However, whenever the medicine is released, anyone can go to the pharmacy and buy the medicine, and they can make their research. That is not so within GMO, because you can only have the seed if you sign an agreement. And it is the company itself which decide what should be put in the agreement. And so when you're a farmer and you sign the agreement, you sign that you will do this and that, and you will certainly not make your own trials, and you will also not give seeds away to someone who would do trials. So you're only allowed to sell it to registered buyers and stuff like that. Some of it makes sense. Some of it is really protection of the company itself. So when a scientist go and approach the company to do research, the first thing the company will say is, but we are very interested in research. However, we have experienced people who were not so good at doing research, who tried to do research, and they came up with some very terrible results. And both for your sake and for our sake, we would really like to protect you against this. So if you present your research to us, the research you want to do, we will assess the prospects of this research you want to do, and then if we think it's okay, you will certainly have the research, the seeds for your research. So the scientist go back and make the protocol and everything that he needs for the research, and he'll send it to the company, and the company will assess it. Now, two things can happen here. One thing is that they say, regretfully, we think this research will not give good results. And so we think you should redesign the research protocol, or we will not be able to release any seeds for your research. So the other option is that they say, we think this is really good research. Now we send you an agreement, and when you have signed that agreement, we are able to send you some seeds for your research. And in that agreement, the scientist has to sign that he will do no other research than that which he has already specified. And he will not hand seeds on to anyone else who will do other research. And certainly this is underpinned in the last sections of the agreement, that this is, if he breaks this agreement, he will have to compensate the company for all damages, including economic damages that the research may do. Now, that was really, when I came to find those articles who told about this, that was when I really started digging. And so I've been digging for the last 4 years, and one of the results coming out of this was that I made this small document. It's about 40 pages, and it tells about the entire range on GMOs. It's regretfully in Danish to you who don't read Danish. I have put a few copies up there, and you can just take them, but only if you want to read it. So whatever is in this one, I will not talk about it today. So what I've been asked to talk about today is more the technical aspects of GMOs. So now my talk begins. So we are tinkering with the DNA, and what I'll try to rush you through is, first I'll summarize it all, so you know what I'll be talking about. Then I'll talk about it, and then I'll hand the word on to Ip. So the summary, we'll have a definition of GMOs. What is a GMO really? And then we'll go through the construct of the GM, the genetical construct that we insert into the plant, the process of inserting it, and the selection that follows the insertion. And then in the end, I'll talk a bit about the GMO black box and the epigenetics that is associated to this black box thing. So when we started out thousands of years ago, we knew what a plant was. Later, we had a microscope and we knew what a cell was. And at a time about 1880, we associated the genetical code with the chromosome. And then we, since 1945 or something like that, we've been exploring the DNA and the double helix here, and we have been able to really understand the genetical code here. And so GMO is really taking this process the other way around. So now we are starting with the code, and we are able to put it into the DNA double helix. We are able to design our genes and put them functionally into the chromosomes, and they'll be functional in the cell, and we will have a genetically modified plant. So we are having the crop that we want, for example here the maize, and we are having the DNA super helix. And a little closer, we will have the plant cell. So we're talking about genetically modified plants. When we work with GMOs, we don't work with plants, we work with cells. And it's the single cell that we transform. And we do that by different methods. I'll come back to those. And one of the important tools here is the gene gun. So they'll simply shoot DNA into the cell. So you can see the cells, and you'll shoot your DNA construct into the cell, and then it'll kind of organize itself in the cell sometimes. So we have here we have a bacteria, and a bacteria like this, it may have the ability to withstand glyphosate, which is the active ingredient in Roundup. So we'll think it very interesting that this bacteria can survive glyphosate. It must have something that can kind of how does it go about not being killed by glyphosate? And so we make an investigation into that, into the genetic code of the bacteria, and we identify a gene. We extract the gene here, we extract and isolate the gene, we grow the gene itself. We are able now to grow single genes and multiply them. And we'll be cloning this gene with other, we'll be making a construct of genes, and we'll be able to put it into the genetic code of a plant, and we'll have that which we call a transformation. So we will have a cell culture. Now single cells, we'll start growing them. Normally when we think of a cell, we don't think of a cell, we think of an organism. What is interesting here is that a cell can grow just by having the right components in its environment. So normally it's the plant or the organism that provides all the nutrients that the cell needs. But today we're able to grow the cell itself just by providing the nutrients that it needs. So it will pick up the nutrients from the medium. And so we are able to have tissue culture, and out of that, with the help of some hormones, we'll have it convert into a living plant. And through plant breeding, we'll have many living plants, and we will cross them with other types of plants, or what you call varieties. So you will have this transformation that you have made. You will be able to have it in a whole range of different varieties. So one event will be used in many different varieties on sale. What I'll be interested in just taking you through is a bit about the cloning process and the cell and tissue culture, which is pretty interesting. Now, definition of GMOs, what is it? The EU definition of GMO, genetically modified organisms, means an organism in which the genetic material has been altered in a way that does not occur naturally by mating and/or natural recombination. Now, it certainly says here, which do not occur naturally. So you could say, even by this simple definition, it is clear that GMO is not natural. So one of the arguments that you hear again and again is that, well, GMO is just like natural breeding. Now we're just able to do it more quickly. But certainly here is a distinction. This is not natural. And in the annex of this EU directive, you can just, if you please, you can make a Google search on this 2001-18 EC, and the directive will appear. And in the Annex 1, this is what is natural. What does that really mean? It is qualified a bit. So when we want to make transformation, first of all we need a stable crop variety. We want to insert the gene or the genes into a variety which is already stable, high producing, high yielding, has a lot of resistance to different illnesses and stuff. And really, as I mentioned, we don't want a crop, we want a plant, and even not a plant, we want cells from this plant. And then we want the functional gene, and it is often from bacteria. And then also, as I'll come back to, we need a gene for the screening of cells, and then we need promoters and stop codons. This is a real genetical construct. We see here we have a promoter and one more promoter, and then we have the glyphosate tolerance, and then we have a gene which gives some resistance to an antibiotic, and then we have more plant DNA here. So this is how it should look on a good day. There are two ways to make a transformation. One is to go through Agrobacterium. Agrobacterium is a bacteria, and it infects cells, and it make the cell give the bacterium excrete nutrients for the bacterium that can live from. So, and to do that, it has a little genetic ring here, and it will try, when it infects the cell, the plant cell, it will transfer this genetic material into the cell, and the cell will then start to do something that to us looked like an illness. What we do is then that we take our gene construct from before, and we put it into this plasmid. And when we then we put the plasmid into the bacteria, and it will not know that it is really transformed. And then when the bacteria next time, and we secure that it does it infects a plant cell, this plasmid with the genetical construct here, it will get into the cell, and we will have a lot of cells that are infected in this way. The other way we can do it is with the gene gun. So we take the gene construct and we kind of glue it onto small gold bullets. And we put the gold bullets into the gun, and we will have a tissue here, and we'll shoot the genes into the cells. This is very common to use the gene gun with monocots like maize. This one is more efficient in dicotyledons like cotton. So we will have the when I'm finished with this, I'll be familiar with this one. So we'll have the gun, shoot the genetical construct with the gold into the cell, and again we'll do it with a lot of cells, and we will culture them, and one of them may show not to be killed when we screen the cells. And so we grow them and we convert them into a living plant. So when you have all these cells that you have tried to transform, many of them will not be transformed, and when we put the antibiotic into the media, they will die. So because we made a construct that both has a functional gene, the glyphosate, for example, the glyphosate resistance gene, and we also put an antibiotic resistance gene. Now, we know that if this construct is functional, this cell should now be resistant towards the antibiotic. And so when we put the antibiotic into the media, like we've done here, all those that are not transformed properly will die. Pretty smart. And then we start to grow each of these cells that are viable in this media with the antibiotic, and we transfer them into other medias with other nutrients, and we put plant hormones in, the right plant hormones, and we will start to form a plant with roots and leaves and stuff like that. And so at a point we can transfer the small plant into soil, and it'll look like a normal plant. Now, we think that GMOs is science. And now I'll show you why it's not science, and why we have all those restrictions, that safety restrictions. One of the good arguments that are regarding GMOs is that the reason why it has not been extended to poor farmers, and we haven't been able to do all the good that we wanted to do with those GMOs, is because it's so costly. And it is because we are having all those redundant security risk assessments. They are not needed. We need freedom from all these rules and restrictions. I just made an example here to show you why it is really needed. We had, let's say, 100 cells that were transformed. 60 of those may not express the new genes, so they are the ones who died in the first, when we put on the antibiotics, because they were not expressing it themselves properly. Then we try to put in the hormones, and some of them will simply not produce proper plants. So we had the cell, and it was okay, but when we tried to put on the hormones, plant hormones, to convert it into a living plant, it may be that it was stunted, or it may be that it had only yellow leaves, or it only had whatever, all kinds of faults that make it a bad plant. Then, when we have the plants, a lot of them will, when we cross them, when we have them flowering, and they'll intercross, and we will have what we expected, viable seeds. And so it may be that some of the seed generation process or flowering process has also been disturbed. So they'll not be able to produce the seeds. It may be that the seeds are infertile. It may be that when we analyze them, it shows that the genes which we know sat there before now are sitting on another gene. So they're jumping around within the genetical code, and then you're not able to use it. And it may also be that they double. So we put in one, and now we have 3. Wow, what will we have next? So because of all these faults that you experience, you started with 100 cells up here, and you may have in the end 5 cells that are really good for a 2nd generation, 3rd generation trials or at tests and stuff like that. And this is not science. This is not science. It is at the best handicrafts, and it is because of all these faults that anyone who knows things about genetic modification, they know that this is the reality of it. So they know that there are a lot of faults in, and there are a lot of genetical faults that can happen when you make a transformation. I'll talk a bit about the one of the promoters. If we go back quickly, here we're having a promoter, and this is also a promoter. So we're having 2 promoters here. One of the most common promoters is called this one. Cauliflower mosaic virus 35S promoter. This virus has 2 genetical sets, and the bigger one of those is this 35 round genetic code. And up here is the promoter sitting, and then we have the genes sitting here round. So all those use this same promoter. And a virus is very strong, has very strong promoters, because plants have been infected so many times. They are used to kind of nullifying invading genes. If they were not able to do that, they would be invaded very easily. So they are very good at pacifying genes that come into the plant. And therefore, when we started transforming plants in the laboratory, we experienced, yes, the gene is there, we can see it sitting in the right place, but it is not expressed. Why is that? And so we found out that we have to put a promoter in front of it, and if we used a cotton, we would put a cotton promoter from the cotton genetical code itself in front of the gene that we wanted to put into the cotton, and it wouldn't really express itself. So they found this virus gene, that virus is really good at invading other plants and animals' genetical code and have their proper genetical code expressed in any cell. Now, the reason why some virus is able to invade this plant or that plant has nothing to do with the code itself. It has to do with the protein coat that it envelopes itself into. So the virulence factor of the virus is not in the code; it's in its coat. So therefore, if you take the genetical code, the promoter in the code, it may really be used in almost any plant cell. Even though this is a cauliflower mosaic virus, it may be used in any cell. And so it's very good at having the rest of the code that is following it expressed. So we used a promoter in most of those commercialized GMOs, GMO varieties that are on sale today. We used this cauliflower mosaic virus promoter. You will also see here oh, sorry. You'll also see here that there's a gene coming up just before the certified promoter here. And that is interesting, because now I'll not take you through this, but just to tell you that here we have the promoter, and this is really the one you should be using. However, handicrafts or lack of handicrafts came in again, and the companies, they used larger promoters than they should have been doing. So all these promoters, this size, and not that size, are used in different commercialized GMOs. And so what? Well, so it seems that the gene that we had here, part of that was taken into of the virus gene, not the promoter, which is not really violent, but the genes from the virus itself was taken along with the promoter and put into plants. And so we see here we have viral replication, we have stability domains. So all kinds of you could call it Trojan genes that followed the promoter are now inserted into commercialized crops. Now I'll just rush you through things, and this is not really just to tell you, give you a picture inside your head of complexity of the genes. Once we thought that, wow, we have the DNA double helix here, and one of those strings is where the genetic code is sitting, and so we will have our messenger RNA made, and it will be transformed out through the nucleus of the plant cell and into the plant cell itself, and it will be converted by RNA and stuff. And it's very, very simple. And then we learned about the histones. Just about the last 20 years, so we really understood, within the last 15 years, we've really begun to understand how complicated expression of DNA, if you have the red DNA string here, and in the cell it's coiled up around histone molecules. And we also see here that we have acetyl molecules, and it will loosen up the association of the histones, so they are easily accessible, also as we heard last night in a very short note. And here we can methylize them, and they'll kind of be compacted and much harder to read. Now, this is the simple stuff we started out with, the DNA double helix, and now we have the histones of different kinds, and we have the DNA code running, and they are more and more condensed, and more and more condensed, and more and more condensed, until they, in the end, is condensed into chromosomes, which they are for a small time of the plant cell cycle. But you see, well, it's just adding up to the complexity of the genetic code. And the complexity doesn't stop there. We have all kinds of systems that we're just starting to learn now. And I don't even understand that myself. So it's just to tell you that the reading of the genetic code is very, very complex. And when we started working with GMOs, we didn't know about most of this. So we didn't know how stuff was really reading, and we're using a gene gun and shooting stuff into this complexity. And then we think we're knowing what we're doing. That, I think, is fantastic. So, because of this transformation of DNA, it may spoil genes in the original DNA code. It may also change the expression of other genes. If you have one gene which is expressed very, very much, like a new gene, you have a virus promoter, so it'll kind of cast a shadow on the next genes on the DNA code. And this shadow of activity will make other genes more active. So while you really thought, wow, we're putting in a genetic code and we're having a promoter, and we thought we knew what we did. What we learn now with genetic DNA investigations into the code expression, we learn that they are casting a shadow of activity on the next genes. So we may have genes stimulated, but others, because of of this acetylation and methylation, we may also have some that are having less expression. Or it may be, as you know, why is it that a root is a root and a leaf a leaf? Why is that? That is because of the epigenetic aspects. That is because some genes are expressed very much in the root, but not at all in the leaf. So that is a part of the epigenetic setup of the plant. And when you change this, when you change the genetic code, you may also change this kind of very strict regulation of the genes. More specifically, I would say that when you have a plant that is able to tolerate salt or water stress, all kinds of stuff, it's able to tolerate or is able to stop some illness of some kind. This kind of stress, it is also a part of the regulation. And you can only see that you have changed it when you see the stress taking on again and you see the plant react as it normally would. So if you don't put this plant onto stress, you will not see that that you change something. We may also introduce unwanted genes, like we have done with the cauliflower mosaic virus, and we may also activate pacified genes. A lot of when you make a transcription of the genetic code, most of the DNA is not expressed. And you'll find bits and pieces from virus and from bacteria and from fungi. Within the plant DNA code, you may say, wow, this piece, it comes from a virus. But it's not active, because plants are very good at pacifying foreign genes. So what's happening here is that when you change the expression of the genetic code, you may activate stuff that really shouldn't be activated and which the plant has, as a routine, kept pacified. And all this we don't know because the authorities do not require this kind of information, and they could easily require that. That was what I had to say.
Hvordan virker det der?